This is episode 110 of the Immunology Podcast, Placental Tolerance and Immunity with Dr. Tamara Tilburgs. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Tamara Tilburgs from Cincinnati Children's Hospital Medical Center on the podcast to talk about her research on immune tolerance during pregnancy. We've also got our usual roundup of recent highlights in immunology news coming up, but first,
this is your bi-weekly reminder that IUIS 2025 is only a few weeks away. And we've chatted about how we're going to get to Vienna, but we'd also like to talk about how you can get around once you're there. The IUIS has partnered with top transportation services to make your journey smoother and more affordable. And you can learn more at www.iuis2025.org/transportation-discounts.
all right.
I know that you're very excited about your stories today. You have a nice two-part complementary set. So why don't you start with that, and then I'll take it on. I'll take it on from there.
Well, and an absolute surprise to literally no one, we have a double header on the microbiome today. I held off and I went to CAR-Ts and Tregs, but you know,
back to good stuff. I see.
Back to the bread and butter today. There are actually two papers in Nature that came out together, so I think they're also doing some type of combined story, honestly.
Mm-hmm.
So I can't tell how many first authors there are, so I am going with what I see is because there's no little numbers on this. But first author is Annie Grebham. Last author is Lisa Mayer. It's in Nature, coming out the 16th of July. Non-antibiotics disrupt colonization resistance against enteropathogens. All right, so we know that if you take a bunch of antibiotics, then sometimes pathogens can appear in your gut afterwards. The most common example of this is C. difficile infection, but other organisms can do this too, right? You break the normal niche and you can get bugs. And so one of the bugs that they use is the Salmonella typharium, or typhus. I cannot pronounce it very well. It's Salmonella entericae, subspecies Entericia cerevar typhimurium. It is basically a standard, it's S.tm, because no one says that all. It's a common organism used to study pathogenicity. It's a gamma proteobacter. So they, in this approach, were like, we know what antibiotics do, but what about other organisms? They looked at 1,197 drugs at 20 micromolar on 5 gamma proteobacteria species in a culture system, and then looked at the responses of those to a 43 commensal bacteria that they'd previously done in kind of this group. And so they looked then for a split, saying, hey, what helped the bad bugs but didn't help the good bugs? And so they found that they used antibiotics as a control, and then ended up with a subset of 20 commensals and 5 pathogens, and found that quite a few bugs, or quite a few compounds affected the commensals, particularly large 3-dimensional volume ones with high molecular weights and hydrophobic, right? So they're getting into the cell wall and causing badness is what they don't say but are saying. And so then what they found is that if you pre-treat with these organisms or with these compounds, then that can cause a bloom if you then introduce invasive species or have the bad bug there as well, and that bad bug then overgrows and causes even death in the mice, in the mouse system, right? And so what they're saying is that the organisms in your intestine are working to create a niche that is protective as a general principle, and even non-antibiotics. You're not just killing all the bacteria, you're altering their function or causing some damage to some of the bacteria, opens a niche. So that's the top line here. they go through the different commensals in this comp 20, which is like a commensal 20 organism set that they use. It shifts gene expressions. So if you do drug treatments, it shifts the gene expression of the organism. And they find that you can have ones that change the biomass. And so you can have a biomass competition, right? So more biomass of the commensals drops the pathogen, less biomass of the commensals enhances the pathogen. So there's the biomass altering category. And there's also then drugs that don't alter the biomass, but have a niche competitor effect, or where like the total amount of bacteria is the same, but like you get rid of one species, and that species is really important for holding in check the pathogen. And so they then added to their set this extra E. coli as one example of a niche pathogen that uses the same metabolic substrates as the pathogen to grow, as the STM to grow. And so if you have a lot of that, the STM is not going to grow, but then some of the drugs really inhibit that bug. In this case, it was a special E. coli's growth. And that E. coli, if you get rid of it because you put one of these, this is like things like simvastatin, right? So these are just common drugs. If you put that on there, not in this case, it wasn't simvastatin. If you put any one of the ones that perturb E. coli, then the drug the pathogen blooms. And so you're finding that the outside of antibiotics, a lot of the very common drugs we take influence our microbiome and then because of that, disrupt it and cause an exposure or outbloom. And there again are three ways. You can either be protective by increasing the biomass of your commensals, just decreasing general commensals, or altering the metabolic niche for them. And so that was the big one from this first paper. This first paper really looked at population effects and, hey, different drugs in different populations and what's changing here. A second paper that was published at the same time started with an even broader screen, but then ended with a single drug they looked at. So this one published online July 16th as well, called Identification of Medication Microbiome Interactions That Affect Gut Infection, also in Nature. First author, Aman Kumar, last author, Andrew Goodman, coming out of Yale. They took a bunch of patient records from over 1 million individuals from 15 years, and then had a control window and a case window, and looked at the odds ratio of a GI infectious event relative to not just antibiotics, but other non-antibiotic drug exposures before that. So some type of data mining they had. And holy Lord, it was impressive. And just in terms of that part. So they found a whole bunch of drugs. They made a list that they share. Cool. They validated these in mice using the same commensal 20 approach, or using a taconic mice that have, specific pathogen-free taconic mice that they pulled in, conventionally raised. So it's not SPF, but conventional taconic mice. To verify it for all these drugs, and they did a washout. What's interesting here is the biggest hit of this all was digoxin, which is a drug used for heart arrhythmias and cardiac contractility. Very common, not super common drug, but pretty well known, very famous drug, been around forever and a day. Nightshade, belladonna, anyone type of, I don't think, is Nightshade the one that is digoxin? It is digitalis, yeah. Foxglove, not Nightshade. Yeah, no, it is Nightshade is a separate family, but it is the digitalis foxglove. So one of those famous, grows in your poison garden drugs. So this case, they're like, wow, Dig had a huge effect. And I'm going to call it Dig because that's what everyone calls it. And they used a strain of STM that's a little less virulent, so they could actually have some data here and not just like have an immediate overwhelming response and kill all the mice. So they go in, they look at this, they discover that, and there's some interesting strain specific effects. I'm going to put those aside because different mice strains are more susceptible to the pathogen apparently. But what they discover here is that DIG pre-treatment or treatment up to 2 days leads to susceptibility. If you treat for 7 days or longer, the susceptibility goes away. So they found it was very context dependent. Now, DIG is known to alter the activity of ROR gamma T, the gene, which is responsible for Th17 cells and ILCs. So they then use knockout mice and look at other things, and through a series of experiments, determine it's the Th17 effect. And so what they're finding is that DIG is downregulates these. So you're having decreases in IL-17 responses as well as IL-22 dependent lectins like Reg3b and Reg3g, which are antimicrobials. And so the defense response available to these mice, if you dose for a couple days, is dropped. So they aren't able to defend themselves, so they don't have the proper Th17 response. See, it is an immunology paper, but it's not the ILCs. And so that is really the key mechanism here. And they do a lot of work dissecting the pathway out, determining the cell types. They then understand that it is not a direct, it's not just the host signaling. So it's not just that DIG is somehow activating a receptor and doing it. So it's not a direct effect on the host. It's actually DIG is affecting the microbiome, which affects the Th17 response. And so here's the plot twist. Short filamentous bacteria are depleted by digoxin treatment. Having enough short filamentous bacteria in your gut milieu is required for proper Th17 priming, which protects you from the pathogen. So DIG kills the SFBs, which are an important spore-forming bacteria. That's bad. And because they're not there anymore with the DIG, although they eventually grow back, apparently, like they adapt to it. But during that hit period, you do not have the signaling that drives proper Th17 responses, and then so you're susceptible to the pathogen. So drug affects bacteria, which affects host, which affects bacteria through the immune system.
That's quite interesting. I mean, I don't doesn't feel very surprising to me. Like it kind of makes sense that this compounds can interact with bacteria in your gut.
It's actually even weirder here. So that does a little bit. So it's even more indirect. DIG binds to ROR gamma transcription factor, alters that. That causes the production of a unique antimicrobial peptide, which then kills the SFBs, which then drops the Th17 response.
Yeah.
So DIG host antimicrobial peptide kills bug. Dead bug turns off priming of immune system to protect.
Yeah. But in your first story, it was more
the first story, it's these big, thick, like lipophilic compounds seem to gum up the bacteria and kill some of them.
Yeah. Yeah.
Even though they're not antimicrobials, they're just big fatty macromolecules.
Yeah. So basically, these two papers are kind of similar in the way that they're looking at compounds that are not antibiotic, but that they are affecting our microbiota in ways that we didn't know.
Yeah, and leading to pathogen susceptibility in particular.
Yeah. Yeah. So when they do for the second story in which they are looking at the clinical record, so basically they see that people that have the digoxin treatment have increased risk of infection.
Yep. Crazy, huh?
Yeah. Yeah. Very interesting. Well, I guess that's important to have a more educated understanding of these interactions, because in the end, you are a whole person. You're not just the heart that gets treated with the digoxin. You cannot avoid having an intestine full of bacteria that will be affected by your modified immune response. All right. Thanks for sharing, Jason. And I'm glad to see that you're back in your element. I hope you're happy. All right. So then I will talk about my stories also, one after the other. And I'm going to start with so I have my stories have also something kind of in common. They're both touching upon well, transposable elements and consequences thereof to some extent. And I thought it was very interesting. My first story, I had to say it's a couple of weeks old, but I saw it, and when I read kind of the abstract, I thought it was very interesting, and I really wanted to share it. I do have a little bit of a bone to pick with this story, so I'm gonna go get to that at the end.
For the
So
record, I want your hot take.
hot take. I mean, what we have to give our listeners, our whole story, our whole read of this. Now I think it's just I'll get back to it. Let's not get ahead of ourselves. Okay, the paper is called Primate Retroelement Exonization and Sexually Dimorphic I13RA1 Transcription Tune Type 2 Immune Responses. First authors is published in Science Immunology. First author Tobias Ploman from the lab of George Casitosis at the Francis Crick Institute in London. And basically, the study is looking into regulation of type 2 immunity. And this is through, particularly in the receptors that are sensing two very important cytokines in type 2 responses, which are IL-13 and IL-4. And IL-13 and IL-4 receptors have some things in common. There's two types of receptors that I'm going to talk about. Kind of type 2 receptors is the most important, which is a heterodimer of the IL-4 receptor alpha gene together with the IL-13 receptor alpha 1 subunit. And this one can signal both IL-4 and IL-13, but it's often not is considered to be not expressed in T cells. So I was looking into this, and there seems to be some situations in which it might be expressed, but as a rule of thumb, it's not expressed in T cells, but it's expressed in almost every other cell in the body. And this is important because it's one of the main ways for IL-13 transduction, signal transduction. And it's important because then on the other side, you have IL-4 receptor alpha. It can also form a heterodimer with the famous common gamma chain, also known as IL-2 receptor gamma, and is expressed and is present in other cytokine receptors. I think also IL-15 has the common gamma chain, or IL-2. And basically, this is the canonical IL-4 receptor that is expressed in hematopoietic cells in general. So I'm going to focus mostly on this IL-13 receptor alpha 1 gene, which is important again for the IL-13 signaling transduction. So basically, one of the big questions in studying allergic disease and understanding type 2 responses is why are, particularly in human health, is why are some individuals more predisposed to developing allergies than others. There's also differences in age. At different ages, there's different kinds of predispositions. And there's clearly, genetic predispositions that we want to understand. And there's also sex differences. And I'm going to get to that at the end of the story. So I want to focus first on one of the observations that tries to, in a way, might explain why some people are more likely to develop allergic responses than others. And in this case, they're studying this gene, IL-13 receptor A1, and there's some polymorphisms that are associated with different, for example, serum IgE levels and a tendency to allergic disease. And it's cool because they're looking at the role of retrotransposable elements, RTEs, that are, inserted in the genome. So transposable elements are these kind of genetic sequences that in principle can kind of jump throughout the genome. There's different types. Retrotransposable elements are ones that can be copied in RNA and then be, inserted somewhere else in the genome. And there's other types. But basically, what's interesting is that this, in general, transposable elements are super abundant in the human genome. There can be like 40 to 50% of the whole genome sequence, which is insane because these are sometimes leftovers from like viral infections. There's some elements that are, very degenerated sequences that I think all of them eventually can be traced back to some ancient virus that happened, long time ago. And the presence and the distribution of these elements in our genome has had such an impact in the evolution of the species. They are known for having, affecting uh, our phenotype in several ways. I think like there was this study that showed that the loss of the tail in primates was a consequence of such a transposable element that cuts some gene. So they are affecting our phenotype all the time. And I think the more we study them, the more crazy they are. And so in this case, there are a few RTEs embed, embedded within the gene of the IL-13 receptor A1 gene. And what they show in what they study, I think it's already kind of shown before, but they really look at it very closely, is that this results in several alternative isoforms of this receptor that can be detected. And they're looking at different types of cells that, because they are expressed in many types of cells, hematopoietic cells, non-hematopoietic cells. So, and they show that they can identify at least 3 different mRNA transcripts, of which one in particular seems to be the most common alternative kind of splicing isoform, which includes a small RTE at the end of the transcript in the untranslated 3' UTR part. And this seems to have very clear differences in the function of this gene. So basically, they see that they call it this IL-13 RA1 LOR1A isoform. So I'm just going to say LOR1A isoform. And this one basically doesn't have the cytoplasmic tail elements. So they don't have these, there are these missing signalings. It has impaired signaling capacity. So the one they look at the structure and they kind of predict the effects of this. But they show that it can be apparent, can reach like 30% of the expression in some samples in some people. They look at cells. I think this is by measuring B cell lines from many different donors. And some people, they don't express a lot, but some people can express a lot of this alternative transcript. And so what they say, they try to understand what is the implications of this difference. And they show that indeed there's having this alternative splicing isoform because of this RTE element. It really affects the function of the whole IL-13 receptor, heterodimer. So on the one hand, you have less expression of the fully functional isoform because it's being spliced differently, but also the protein itself outcompetes, if expressed high enough, it can outcompete the good isoform sort of, and therefore this reduces in the end the signaling of IL-13, and this has effects on the, for example, IgE production by B cells expressing these isoforms. So they have a lot of a lot of models in which they show this. So basically the inhibition because of the expression, the competition, and they show that in part this is because of differences in the internalization of the receptor when it binds to IL-13. And this kind of, to some extent, could explain some differences from person to person in the tendency to, for example, produce IgE in an allergic response. And then they have a second part in the study, and this is the part I'm a little bit a little bit confused about. They also are looking into sexual dimorphism in the expression of these of like IL-13 receptor, and basically they show they find that there's higher expression of IL-13 receptor A1 in males compared to females, but this only happens in individuals of European ancestry. And it's interesting because we already know that there are some genes that have different expression. I think TLR7 is a well-known example that is more highly expressed in females because of it's expressed in the X chromosome, and therefore sometimes you don't have uh, um, complete silencing of the second X chromosome in women. Therefore, you have a higher dose of the TLR7, and this has been associated with increased autoimmunity or allergy in women. But what I'm a bit confused is that in this study they are implying that men have higher rate of allergy because they have higher expression of this receptor, and that's something that I'm actually a bit confused because I also understood it was the other way around, that and so they show that it is to some extent the estrogen signaling is repressing the transcription of these IL-13 or RA1 receptor, which would mean that women, after puberty and before menopause, according to this, would explain lower incidence of allergy, which I don't think is the case. I understand that that's actually the other way around. That women, that although boys and before puberty, there's higher incidence of allergy in males. This is reverted in after puberty. So I'm a bit confused about this, but basically they show that there's estrogen signaling reduces the expression, which should correlate with lower incidence of allergy. So maybe that's something that I'm misunderstanding, or I'm outdated in our current understanding of allergy in
You
males
are
and
you are
females,
you are not.
right? So I'm a bit confused because they actually are looking. They really make the point. They say that the sex difference. So this is like the sex difference in IL-13 or when expression was more pronounced in females before menopause, but not in older females. So it's really supporting this, and estrogen is mediating this.
Yeah,
But then,
sex hormones mediating allergy is 100% the case. It
yeah,
could be that you have competing things that any hormone does pro and anti-allergy, and this
yeah,
isn't enough to shift the story based on other things, perhaps.
yeah, yeah. So I guess I'm a little bit confused about that, but maybe the authors can comment on this and show where I am misunderstanding their beautiful work. But in any case, I think this is really nice because we're using understanding the role of these transposable elements in our genome, which they're there. We gotta deal with them, and also I think it's always very important to start understand sex specific regulation of certain immune elements because this means something to the patients and to the phenotypes that we see in actual people. So kudos to the authors, and yeah, just if you wanna correct me or add something to this, please make sure to reach out to us. And well, I guess that I should the risk of making this a monologue, I'm going to talk about my second story. I'm trying to be a little bit more concise. And the second paper for today is called DNA hypomethylation traits define human regulatory T cells in cutaneous tissue and identify their blood recirculator counterparts. And this is also a bit, up my alley, going for the T-Rex. Paper was published in Nature Immunology, 16th of July. First authors are Niklas Bäumer, Charles Imbush, and Tamara Kaufmann. From corresponding authors Michael Delahair at University of Mainz and Markus Feuerer at the Leibniz Institute for Immunotherapy in Regensburg, Germany. And in this story, basically, they are this labs are very well acquainted with regulatory T cells, and I think nowadays it's really hard, but we are trying to understand the role of T cells, not only that we find in the blood, which is kind of the low hanging fruit, but actually understand the role of Tregs that are in the tissues, because we know that uh, Tregs in tissues are very important for the homeostasis and the normal functioning of tissues in general. So we know we have clear population of regulatory T cells in the skin, in the adipose tissue, in the muscles. And I've been talking about stories around this for a long time, and I've been reporting this from our conferences. So it's a very interesting, very well studied topic at the moment. And but of course, it's hard to get tissues from humans. You can always, or make meaningful experiments with T-Rex from humans. So one of the things that people are doing now, because we have all these tools and very sensitive assays that we can do, is looking into methylation patterns and how they relate to gene expression, to chromatin accessibility. And this is important because we know the methylation I use is usually a bit more, it's easier to, how to say, the methylation patterns are a bit more sustainable, a bit more durable. And this to some extent allows us to understand certain relationships between cells, where they're coming from, where they're going, because methylation patterns stay on the cells for longer than just gene expression that changes as soon as the cell leaves the tissue. We might just lose that expression that we use. So in this paper, they show, it's really also very, I can imagine this has been a lot of work. They're looking into methylation patterns, chromatin accessibility, and gene expression of many, many Tregs from many different places. From tissues, they're focusing mostly on the skin, and they look at different populations of the Tregs, and they compare them to Tregs, different populations of Tregs from the blood. And there is one receptor that really stands out from this analysis, which is CCR8, which I forgot which chemokine is supposed to be responding to. But CCR8 has been shown to be very upregulated in, I think most of the interest in CCR8 in Tregs started in the cancer, business, because it is a very commonly expressed receptor on Tregs that are found inside tumors. And we know, of course, that Tregs in tumors are probably a bad idea. They're probably inhibiting or preventing the immune response in the tumors. And so people are really thinking, well, can we deplete CCR8 positive cells and then, remove this layer of suppression in tumor? Would that help the immune response? That would help, tumor clearance. But then we know that there's also this population of CCR8 cells in other places. We see them in blood. There's a small population. And CCR8 also seems to be expressed on Tregs in the skin, in tissues. And this is kind of where we try to understand what are Tregs in tissues and where are they coming from? What are they doing? Do they circulate? And what are the risks of, targeting CCR8 when thinking about tumor Tregs? So they show that in this paper, they look into this methylation, and they establish certain signatures and methylations. In the methylation, they look very genome wide. They're looking very into many, many methylation sites. I think it's like 28 million CPGs that they analyze, and they compares subsets from the skin and the blood and the CCR positive Tregs in the blood, and they really find certain. Basically, what the first thing they find is that in skin Tregs, there's much more hypomethylated sites than in the blood, than naive Tregs from the blood. So the methylation landscape is completely different, and it's in general much less methylation. And they find specific clusters of CPG, hypomethylated and non-methylated CPG that are different between this skin naive Treg in the blood and CCR8 blood Tregs. And they really develop these kind of signatures for skin, for conventional cells also in the skin. So they kind of compare to the conventional cells in the skin. And the most thing is there's this core naive signature that is what you expect Tregs to do. But then there's this hypomethylation signature that is kind of closer between Tregs in the skin and conventional cells of the skin that really, I think, is a kind of tissue residency signature. And so there's also they compare, so because they also have data on chromatin accessibility and expression. They have these kind of what they call DMR peak gene triads, in which you look at the three things and they see how this correlates. And they show that indeed demethylation of certain regions relates to more open chromatin, more accessible chromatin, and with upregulation of several genes. And I think that interesting here are, CCR8 is within these genes that are in that seem to be in areas that are hypomethylated and therefore are more expressed. They have some, for example, they have other kind of activation of genes that are related, that are often observed in tissue resident cells, and they have kind of hypermethylation of, for example, CD62L and TGF beta receptor 2, and those are more silenced, and they can also see that on the transcription level. And basically, they see enrichment um, in certain pathways that are associated with skin residency. They find also certain transcription factor motifs, and they have specific transcription factors. And I want to say June and FOS, for example, important that are downstream of T cell activation, and they have other ones that also show demethylation, and so increase this one, so demethylation and increased accessibility. And then they have others that show demethylation, but they're not necessarily more accessible. And this idea of having changes in methylation or changes in accessibility, I struggle a little bit to kind of explain this, or like to kind of understand the relevance of this. But so accessibility and hypomethylation are not necessarily together, but you need, in a way, both to have increased gene expression. Methylation is more permanent than just accessibility. So they show that for some cases you have both, but for some cases you have the demethylation, but that doesn't mean that you have increased accessibility. And this is what they see in the CCR8 positive Tregs in the blood, is that in general, they are very similar. Their methylation patterns are very similar to the ones in the tissues, but not their accessibility or their expression, gene expression. And the way they interpret this is that you have this as the Tregs. What they propose is that you have the CCR8 cells, Tregs in the blood, are recent emigrants from tissues, or they are kind of circulating from one tissue to the other. And this is something that has been recently also shown in Tregs in mice, but it's harder to look into humans because you cannot do parabiosis in humans. But it's
shouldn't do parabiosis.
You shouldn't do parabiosis in humans. But then the idea is that using this methylation, understanding the differences between methylation, accessibility, and transcription, is that they can really support the idea that CCR8 does mark Tregs that have an intimate relationship with the tissue. They have recently left the tissue, and they're probably going to go back to the tissue. But it is the methylation where you see most of these durable signature. And they show also they do clonotype analyses, and they also see a lot of overlap in the clonotypes, the CCR8 positive cells and the cells that they see in tissues. But I think this is in mice. And in mice, they also look something that you can do in mice. So look at a photoconversion. They have these mice that if you they have a photoconvertible fluorescent protein that switches from green to red if you expose them to violet light. And so then you can mark them in the tissue and then see whether you can find them back in the blood. And with it doing kind of combining all of this, they really conclude that the CCR8 cells, Treg cells in the blood are closely related to the ones that are found in the tissues. And I think this is very interesting because, then it gives you a glimpse to the tissue that you can actually find in the blood. And just as a last thing, talking about transposable elements, again, transposable elements, I mentioned they are very abundant. They're everywhere. So they're also looking, because they do this very detailed methylation analyses, they show that hypermethylations these transposable elements are actually are disproportionately enriched for hypomethylations, and they are really very much affected by hypomethylation patterns in the tissue resident cells. And they also, in this conserved hypomethylation CCR8 positive cells. And they see upregulation of certain RNAs that are, because hypomethylation can result in upregulation of the expression of these transposable elements that are usually silenced with methylation. And they don't go as far as to say exactly what is the consequence of that, but these hypomethylated transposable elements are present in many of these skin Treg signature genes that they mentioned before. Just TNSFR8, which is, I think, is CD30, PREX1, not familiar with that one, PRDM1, BATTF. These are all kind of already been prescribed. And so the question is whether is this hypomethylation affecting the expression of these genes through, for example, targeting these transposable elements that we tend to kind of not think too much about, but maybe we should be looking at them too, where we look at methylation, because they might be actually affecting the expression of genes in a way that, yeah, it's it's durable for one, so it gets, it lasts in time, and might be affecting the expression in ways that we weren't completely aware of.
Yeah, so I was trying to, at the first part I was like, oh, methylation sites and DNA accessibility are there's patterns for, say, the skin that are inherent, and that predicts that it's in the skin. I'm like, you would expect that, right? Since it's not actually technically a different genome, that it would be accessibility driving your
Yeah.
expression. Like that doesn't like surprise me. But I guess what's interesting is the relation of some of these cells and the resident to the floating. But I think we've seen that story before too, right? That like what we see in tissue versus circulating, like
Yeah.
it makes like a copy that's just a little different, decides to leave home, kind of like your kid.
Yeah.
So
So I've spoke about this before, but it was mostly on mice. There has been a study looking into recirculation of Tregs in mice and how they move throughout the tissues from the blood and they go back to the tissues. I think it's harder to do in humans. But I think with the hypomethylation, I don't think it's necessarily obvious that you would have a general hypomethylation because of tissue residency. Um, I
I mean,
think that is,
how else is it going to be controlled? If you need to permanently alter your transcriptome profile, you're going to do some of that.
yeah, but why hypomethylation? Why not hypermethylation? Why not
Sure.
silencing genes instead of activating?
I guess that's a new part, but like the fact that your chromatin accessibility is altered in some way.
Yeah. Yeah,
Yes.
that makes sense.
Yeah, like that's what's supposed to happen, right? Otherwise we're kind of in weird science space.
Yeah. 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 Um,
Well, that being said, we're going to be speaking to Dr. Tamara Tilburgs at the Cincinnati Children's Hospital in just a moment. But before we get to that, have you thought about how long it takes you to isolate immune cells for your research, Brenda?
all the time, Jason.
Brenda, would you like to speak things up?
For sure. Tell me how.
Well, EasySep is a column-free technology that allows you to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Immunologists around the world choose EasySep to isolate immune cells and accomplish more with their time in the lab. Learn more at stemcell.com/chooseeasysep.
So now we move on to our conversation, our interview today, and we're really happy to welcome Dr. Tamara Tilburgs. She is assistant professor at the Department of Pediatrics at the University of Cincinnati. And I'm really happy she's talking to us today because her labs investigates studies maternal immunity and immunity during pregnancy. And I first saw one of her lectures during IUIS a couple of years ago, and I thought it was such a fascinating topic, understanding how the mother interacts, with the placenta, how the antigens from the father get tolerized, and I think it's such an interesting topic to talk, maternal tolerance to during pregnancy, and I'm really happy that she's discussing it with us. So, Dr. Tilburgs, thank you so much for joining us for the Immunology Podcast.
Thank you, Brenda. Thank you so much for having me. I'm really excited to talk about this topic because I know it's a topic that we as immunologists don't always think about. So I think this will be a wonderful conversation to highlight that.
Yeah, I think recently I've been thinking a lot about maternal immunity and how we live with women live with a whole human being growing inside of them. I think that's really really interesting. And I think maybe if you allow me, Tamara, we can start a little bit describing the problem. So, or maybe better said, the unusual situation in which happens during pregnancy, in which you have a fetus that has different genetic material compared to the mother. So how does this work, and what have we learned in the past years about this interaction between under these conditions? What do we need to know?
So I think I can summarize a little bit of what we did, and also be really excited that our field actually is growing. And I think a lot more emphasis is placed on both how during pregnancy, the mom is accepting this foreign entity in her body and how she tolerizes for those paternal antigens. Because I think we can learn a lot about like how the immune system actually works, since pregnancy is like essential for evolution and survival of the species. So my work has really always focused on human pregnancy, and in the interface, in the uterine tissues, the placental tissues, there where the maternal immune cells actually can recognize these immunologically foreign cells and tissues. So the placenta really forms the interface between the mom and the baby, and it really provides all the nutrients and gas and oxygen to the baby. But there's loads of different structures, and the cells that are extra embryonic are called trophoblast cells. And the trophoblasts, I think, are actually where the immune paradox lies, because there are several types of trophoblasts that are very invasive. They invade into the uterine tissues, into the maternal spiral arteries. And it's those trophoblasts that actually express MHC antigens. So HLA-C, not A and B, which is also very interesting. We do not understand how they come to express only one HLA-C type, HLA-C, and not the others. But we think that that is to limit their immunogenicity and have a little bit less of this antigenic load, while HLA-C can then still provide immune protection in case there is an infection. And I think what our lab really has been doing for the last 5 years here in Cincinnati, but before I was in Boston and in the Netherlands, is really trying to first define how are those maternal immune cells that include T cells, NK cells, cytotoxic T cells, or regulatory T cells, how are they interacting with those trophoblasts that have HLA-C? How does that establish tolerance? So how does that increase regulatory T cells? How does that prevent cytotoxicity? But then at the same time, what mechanisms of immunity are allowed there? So that in a way that doesn't jeopardize the placenta and cause rejection. And then currently we're taking that a step further, where we also want to look in placental inflammation and pregnancy complications, especially lesions like chronic chorioamnionitis. It's an inflammation of the membranes, but also chronic philitis, which is an inflammation of the placenta, where you see actually massive T cell infiltration, CD8 T cell infiltration into these placental structures. We have no idea what is those T cells are seeing, what kind of antigens are triggering this. We don't know why they suddenly breach all these barriers and cause a lot of trophoblast cytotoxicity. So I think there's a lot of focus in my lab and also by others to do both, to understand the drivers of pregnancy complications, because these lesions are really important for developing a preterm birth or weakening of membranes and premature rupture of membranes, fetal growth restriction. So all kind of really frequent pathologies that we see in pregnancy for which we do not have any therapeutics. So I think that's really like a need in our field, to really understand the drivers so we can provide solutions. But at the same time, we're looking at how are those T cells and NK cells and trophoblasts talking to each other in healthy pregnancy, and learning about all the fascinating ways that they can still kill bugs, kill viruses, kill bacteria, a little bit differently than anywhere else in the body, because they're always trying to avoid rejection and excessive inflammation. So I don't know if there's any like particular cell types you would like me to talk about in more detail. Like I love all the cell types. That's kind of fun. And I think kind of unusual for a lab. But we literally work together as a lab, where one grad students works on NK cell, a fellow works on trophoblast, somebody works on CD4, somebody works on CD8 T cells. So when we get these tissues, we can all get our cells and work together to process these materials and kind of like get an understanding and integrate also a lot of the data.
That was actually my question: is which of we think immunosuppression, how central are Tregs and MDSCs, the classical two big ones, to the processes you see with what the placenta does here? Is that typical? Is it different? Is it normal? Or is there other stuff that we don't really understand going on?
So the regulatory T cells, I've not worked too much on dendritic cells, mainly because there's very few of them in the placenta, and we think that the major antigen presenting cell are actually the macrophages, and there's just few DCs. But we've done a lot of work on regulatory T cells, and we've actually shown already from my PhD in Leiden University that there's a lot of Tregs in the placenta. We have some evidence that they seem to migrate from the peripheral blood into the placenta, into the maternal sites of the placenta. Later, we discovered that trophoblasts can actually directly increase FOXP3 expression in T cells. So they're really like promoting Treg expansion and function. In a Cell Reports paper from 2019, we actually identified additional types of Tregs. One is a PD-1 high IL-10 secreting Treg. It doesn't have FOXP3, so it's not a natural Treg, but it's very suppressive. It uses IL-10 to suppress CD8 T cells, and that one is actually my favorite, because the interesting thing is that this extravillous trophoblast that only has HLA-C, it doesn't have class 2, needs direct cell contact with a CD4 T cell to expand those PD-1 high IL-10 secreting cells. And we have no idea. It seems to also be blocked by CD3 and TCR interaction. So there seems to be some specificity going on, and we're trying to really elucidate the mechanism. Like, how is TCR involved? How does that work when you have a class 2 negative trophoblast that only expresses C? And then what type of supporting mechanisms and molecules enhance that PD-1 and IL-10 in those cells? So yes, I think the interesting thing, though, if you look at mouse models, blocking Tregs really does cause the demise of the pregnancy as well. So I do think that establishment of tolerance by increasing Tregs is essential early in pregnancy, in mice, in humans, across species, to really allow those trophoblasts to grow and form that placenta.
So I have so many questions, but so you mentioned Treg, so maybe we could just take a step back and say, in a normal placenta, of course, trophoblasts are basically what is making the placenta, but what in a normal successful pregnancy, I don't know how much data we have on that, because by definition we wouldn't be touching those tissues, but one is the normal situation. So we say influx of Treg seems to be normal. Do
Yes.
we have also other lymphocytes? I think I remember NK cells also have been well described. You mentioned CD8 cells, but I guess more in a more pathological situation, or also in the normal situation.
So when you look at the cross-section of the decidual tissues and in the interface, so the decidua is actually the endometrial tissue that turns into the maternal side of the placenta. So we think that some immune cells remain throughout pregnancy and some can come in. But actually in a healthy pregnancy, and this is mainly based off collecting placental tissues after birth. So when we take healthy births with no complications, you can dissect those decidual membranes and the implantation side, and you see tons of Tregs, but actually the dominant population of CD4s is a tissue resident and an effector memory population. When you trigger them, they actually make tons of pro-inflammatory cytokines and also granzyme B. There's tons of NK cells. NK cells are most dominant in early pregnancy, when they're about 70 to 80% of pregnancy or of decidual immune cells. But at term, they still remain between 20 to 50% of immune cells. So they are very large fraction of immune cells. They're loaded with perforin and granzymes. We think that they're real immune effector cells to kill microbes. And then there are CD8 T cells, which I think are fascinating. CD8 T cells come in lots of different flavors. We published a JCI Insight paper in 23. And we know that there's virus-specific cells. We know that there's fetus-specific cells. The CD8 T cells come in a lot of flavors. Again, effector memory, effector cells, resident memory cells. The resident memory cells have a lot of variation in how well they can degranulate and produce pro-inflammatory cytokines. But the effector memory cells, again, they make tons of TNF alpha and interferon gamma. But the interesting about the CD8 T cells in the placenta is that they're blocked from making perforin protein. So they have a lot of RNA for perforin and granzyme B, but we have a new study going on, and it's not published yet, but I'm happy to talk about it, where we think that the trophoblast is actually informing the CD8 T cell to not make perforin protein. So there's a real, and we think it's soluble factors that are causing a block in perforin production. So the trophoblast does not do this to the NK cell because the NK cell is allowed to make a lot of perforin. But interestingly, when we have like infected extravillous trophoblast, the NK cells never kill them. But the NK cells are pretty good killer cells. When you add a maternal stromal cell, then they're as effective as PNK in clearing infection. But as soon as it includes trophoblast, they seem to promote Tregs and shut down cytotoxicity at different levels in healthy pregnancy. And we're trying to obviously now get at if there's any dysfunction of these processes present in these inflamed placentas.
So do you have any mechanism about how they're doing this?
That's going to be the final push that we're making for the paper. We're doing loads of RNA sequencing with and without trophoblast to see where the block actually falls. I think it's a real translational block. So maybe induction of microRNAs, something like that. It looks like TGF-beta plays a role, but TGF-beta plays a role in everything, and it's not the only thing. So yeah, we're working on that. I hope that I hope that by the end of the year we have some more information on the mechanism driving this.
Yeah, it feels, if I'm going to be very naive about it, it has to be wrong. There cannot be any, cells that can make inflammatory stuff next to the placenta. That has to be problematic. I don't know. That's kind of my first feeling is you don't want inflammation next to this very, delicate thing growing in there.
Sure. So the way I see that actually, because I think that's actually the first response a lot of people give, pregnancy is kind of seen as this state of tolerance and immune suppression because everything is there to tolerate that foreign entity. But I think it's actually a state of inflammation. And then inflammation really serves different things. The immune activation, and Ashley Moffat describes this really nicely, is also needed for tissue remodeling and trophoblast invasion. So you need to produce lots of growth factors and cytokines for tissue remodeling. But what we're focused on is that I think you need those immune cells there to prevent infection. Infection, in any case, is detrimental for pregnancy. It's associated with preterm birth, fetal demise, stillbirth. Luckily, now we have vaccines and antibiotics. But if you lived in a society 200-300 years ago, you wouldn't have access to this. And then infection is a major driver of maternal and child death. So I think what these cells are doing there is providing immunity by probably killing infected cells of maternal origin very effectively. So that's what the NK cells can do. I think that's what the CD8 T cells can do as well. But as soon as that so they're trying to keep the spread from the maternal tissues to the placental tissues. And by that, they're keeping the placenta clear of infection. But once the infections reach the placental trophoblast, then they're much harder to kill and target. There's alternative mechanisms which don't work as effectively, We have one story in Cell where we show that NK cells make granulysin, and it's really an interesting antimicrobial peptide. And what the NK cells do is they specifically transfer it through nanotubes into a trophoblast, and then that granulysin is used without perforin and granzymes. It kills the bacteria in the trophoblast, but it doesn't kill the trophoblast. Whereas normally, if this would happen to a maternal cell, it would be using perforin, granzyme, and granulysin. So then the maternal cell and the bug would die. But in the trophoblast, again, the whole system is there to allow some immunity, but to always prevent cytotoxicity of the trophoblast. But I think the immune activation, again, in summary, is there to provide immunity and to keep all those microbes out. Because as soon as you have microbes in the placenta or transferred to the baby, you are in big trouble. And then also both the mom and the baby have a very high risk of mortality.
So how does, if there's any key principles, how does the placenta understand, host infected, mom infected badness, kill, versus foreign antigen baby, don't kill? Is there like some core principles you've been able to understand that really lock in?
So I think there's always a don't kill signal. There's, even when we infect these trophoblasts with cytomegalovirus, with listeria, with E. coli, the NK cells and the T cells don't kill these trophoblasts. They're very resistant to killing. Now, the placental structures do have TLRs and NLRs. So they're intracellular and extracellular sensors of microbes. So we haven't done a lot of work on them. I think they've been more studied in mouse models, but I think the NLRs were quite different between humans and mice. And again, placenta is always slightly different than other tissues. So I think we need to look at the sensing of both of the trophoblast and also of the dendritic cells and the macrophages, because they would be the first responders. But I think when we think about T cells and NK cells, they still do not kill the trophoblast, even when there is a listeria, E. coli, CMV, herpes infection of those trophoblasts. They kill when the maternal cells are infected. So I think, yes, that's where we need to go. That's what we need to understand, to look at more information from cells that come from infected placental tissues or severely inflamed placental tissues, and see which sensors were activated and what is driving that inflammation.
So you did a lot of your work. You're focusing on samples from human tissues, because it's difficult to model, to study pregnancy in our typical animal model, the mouse, because it has different structure. The structure of the placenta is different. Do you want to maybe also explain to our listeners what are the challenges in studying this system in animal models?
Yeah, I think there's a lot of challenges. I mean, there's also some advantage, of course. So I think there's a place for using animal models for pregnancy. But the main difference is that humans, gorillas, and chimpanzees are the only species that have HLA-C and HLA-G homologs or orthologs. So mouse models don't have much class 1 on their trophoblasts in the first place. There's might be a few stem cells that have some class 1. So there's a lot less of this direct immune recognition by KIRS and TCR that does just not take place in the mice. Also in mice, there's very, very few CD8 T cells. There's a lot of Tregs, and when you deplete the Tregs, you do get resorptions. But you need an infection to get the CD8 T cells into the placenta. Whereas in humans, there's tons of them already there. So and then also gestational length, 3 weeks versus 9 months is a very big time difference in terms of memory development and responses. I think there's a lot of good work being done on the macaque model, which is a little bit more similar, even though they have, we don't know if they have any class 1 on their placenta. They don't have HLA-G homologs. There might be a soluble HLA-G. HLA-G is super important for us to identify extravillous trophoblasts, so these invasive trophoblast types. And I think it's one of the key drivers of this tolerance. So we can't study that. So I think we've recently also, we just got a paper published, accepted, where I do collaborate with a group in Minnesota from Nathan Schuld that uses a mouse model where they give it's a dirty mouse model. So it's a mouse that are co-housed with a pet store-bought mouse. So they get like a natural microbial exposure. And in those mice, we actually find a lot more CD8 T cells in the placental tissues, probably driven by that exposure. So that might be a little bit of a better model for us to study CD8 T cell homing and differentiation in placental tissues than the normal housed SPF mice. But I think we still have the problem that there's mostly indirect antigen presentation and that there's not really a lot of direct MHC recognition, which is so unique for humans. Yeah.
Do you think that's by design, though, in a way? Like that is how you don't have immediate placental host fetal issues?
Exactly. And I think every species has found its own solution for dealing with pregnancy and infection at the same time. So every species has different types of invasive trophoblast, different types of KIR, different types of T cell responses. And I think the human situation is so unique. And I love the philosophical aspect of thinking about this. But because we have such big brains, in the 3rd trimester of pregnancy, the placentas actually need to transfer a ton of oxygen to the baby just to facilitate the growth of the brain. So by the time after 6 months, the placenta needs to be super efficient in getting all those nutrients, all that oxygen across. And that's been hypothesized why we have such deep invaded placentas that are super functional in transferring that oxygen. But I think also that deep invasion makes our placentas a bit more susceptible for hematogenous infection because it's just more opened up than a mouse. And I always say, like, evolution doesn't care about the individual. Evolution cares about the species. So I think there's definitely different solutions between animals, but I also think that there's different solutions from every pregnancy to every pregnancy, how it deals with mismatches and infections. And I think we're also really trying to learn actively from that to see within healthy pregnancies. We know some pregnancies have tons of Tregs, others actually don't, and these are all healthy. So we're trying to understand the donor to donor variation and how these different solutions are driving healthy pregnancy, so that we can also recognize what is actually more pathogenic.
So we're talking about, of course, protecting from infections, and that's pretty kind of, I think, conceptually is easy. You don't want anything bad to happen to the baby. Let's talk about understanding the effects of the maybe immunity against antigens that are present in the placenta, which is the point of contact. They are from the mother and therefore are foreign. And what happens when this goes wrong? What is our current understanding about when things go right and when things go wrong in terms of that?
Yes.
It must be hard to study, but what do we know about this?
It's quite hard to study, but I think we have quite a lot of data here. So we do know that there is recognition of these paternal antigens. 50% of women actually actively make cytotoxic T lymphocytes to the Y chromosome. In response, so that obviously women don't have that, the baby can have it, and this is not detrimental to pregnancy, because 50% of women make that when they have a first boy, and then up to 80% when they have multiple boys. We know that you can have antibodies for any HLA that your baby has, including HLA-C. That doesn't seem to be really driving pathology either, even though HLA-C antibodies might have a slight higher risk for miscarriage. But in generally, there is a high frequency of women who make these antibodies. We also have a cohort from my PhD work in Leiden where we compared moms that were mismatched versus that were matched on the HLA-C locus. And the interesting thing there was that the CD4 T cells were definitely more activated when you have a mismatch. There were also more regulatory T cells when you have a mismatch. When you have no mismatch, there seem to be fewer T cells as well. So from there, we postulated that there is immune recognition of that foreign HLA-C, but that immune activation drives tolerance and not cytotoxicity in a healthy pregnancy. Now, in terms of, and we've confirmed this with additional studies, including that we think that HLA-C is driving PD-1 high Tregs. So I think it's an active induction. It's an active recognition and induction of tolerance, even though parts of this story we still have to really prove, and it's still an hypothesis, a working hypothesis. We also know that when a trophoblast has a lot of HLA-C, it also has a lot of PD-L1 to every inhibitory molecule that you find in cancer. So anything that has high HLA-C has a lot of immune protection. Trophoblasts that have low HLA-C or low don't have much immune protection. And it's the same with donors. We have a cohort of about 25 donors now. There's donors that have super high HLA-C, and they generally also have loads of PD-L1, PD-L2, PVR, again, like GARP, like probably like 12 different types of immune suppressive cell surface markers that they make actively. Whereas donors that have low HLA-C expression on their trophoblasts don't make a lot of those proteins. So again, I think the data and the story that we're working with is that I think in healthy pregnancy, this HLA-C is being actively recognized. And because there's expression of all that tolerogenic environment, it drives tolerance, both on increasing Tregs, suppressing CD8 T cell perforin, not allowing cytotoxicity. So now we want to obviously take a step further and not only look at healthy pregnancy, but look at the pregnancies with chronic chorioamnionitis, chronic philitis. Those pathologies really have 100-fold more CD8 T cells in their placental tissues, even in the villi. They breach the barriers. And there, we do not know what is driving this. We don't know if that's a microbial response, a viral response, an anti-HLA-C response. But this is actually where I got my recent NIH R1 awarded for, to really trying to figure out what are those CD8 T cells seeing on that trophoblast. We know that there's a lot of trophoblast death in those membranes and also in the villi. So we really think, and we see synapses, we see actually CD8 T cells making a synapse with a trophoblast. We still don't see a lot of perforin in those synapses. So I don't know if it's a classical perforin-dependent killing of those trophoblasts, but I think our plan is to create a cohort of placentas that have this lesion versus placentas that don't have this lesion, and then really do both in-depth phenotyping of the T cells and the trophoblast, both on the protein level, which I think we need because there's a discrepancy. A lot of discrepancy between protein and RNA expression, but then at the same time, we'll get single-cell RNA-seq data, clinical data, pathology data, cell count data, and then using methods developed in computational cancer biology to trying to find out what are patient subtypes within these lesions, and can we find groups of these placentas that have possibly a really strong antimicrobial or antiviral response. Maybe others look more like a rejection response. And I think really like defining that is going to be essential to get real optimal therapeutics, because if we need to suppress a anti-rejection response, it's going to be very different than if we need to enhance immunity and clear infection.
Well, I'm sure we could gestate on this conversation for a full 9 months. Sorry for the terrible dad joke. It was just right there. I had to go for it. But we are running out of time. So at the end, we asked kind of like a spontaneous, fun question for people that's not necessarily related to their day job. So for you, if you could have one superpower, what would it be and why?
Oh my God, a superpower.
Not of the comics.
Yes. Well, if it was for science, I would like the superpower of that my brain is a computer and can hold all these different types of information in my head and process it like AI can do and make all these beautiful graphs, because I think I love data, but your brain is so limited in thinking across dimensions, and you have to rely on computers to tell you, and then you have to do all these sanity checks. So I think I would like to have an AI computer in my head that I can trust.
Superhuman intelligence.
Yes.
That sounds like it could be good, but then you have to be able to like switch it on and off, otherwise it would go crazy.
Oh, totally. I would only use it for science. I wouldn't use it for the stock market or anything else.
And I guess on a related note, so you use it for research. And so another question we have for you. So when you're not conducting research, what do you like to do, on the side?
So when I don't do research, I actually spend all my time in my pottery studio. So I used to do always abstract painting on Monday nights during my PhD and throughout my life. And then in the pandemic, I switched to ceramics. And I think it's just the best way of disengaging my science brain, meeting some other people that are normal people, not scientists, and then resetting my brain so that the next morning I love science again and I don't get burned out.
That sounds nice. That sounds nice. Some pottery, some creative endeavor.
Exactly. It's not easy. You have to spend time, but it's really like, it's my therapy. It's like, if things are hard, like I go to the pottery studio, it always makes me feel better. And I think it also gives me more like brain space to make creative solutions for scientific problems. So I love the combination of science and art. Yeah.
Awesome. Well, thank you very much for coming on.
So thank you so much to Tamara Tilburgs from the University of Cincinnati for talking to us about maternal immunity in the podcast. And we're hoping to maybe see you soon at a conference or something like that.
Absolutely. And thank you so much for having me and talking about the most interesting topic. Thank you.
Well, we are at the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.